Shannon again. So we're going to continue on now with our genetics portion of our notes here. So I am on slide 51 of the in-class notes, and I'm going to go ahead and hit present mode, and we're going to get started. Okay. So we don't spend a whole lot of time on being able to look at phenotypes, genotypes, and predict phenotypes and vice versa. We spend just a couple of weeks on Mendelian genetics and non-Mendelian genetics. If you took genetics with Mr. Hipschman or Mr. Ship, you would have spent a lot more time on Mendelian genetics, probably an entire semester. We're more interested on predicting inheritance patterns and doing general probabilities than we are in looking at a lot of different examples of each type. You will want to have a couple of examples in your toolkit just to use if you have an FRQ, for instance. So Mendelian genetics is based on the research by Gregor Mendel. He was a monk. He studied plants, which a lot of our earlier scientists, this is what they did. He looked at phenotypes. He looked at physical characteristics and came up with ideas and different theories and laws about inheritance based on these phenotypic traits. Now, remember, just as we're talking about genetics, that we're talking about genes and alleles of genes that come from both sperm and egg from each parent. We have a copy coming from each, so we have 2 copies in total. So really quickly, some vocabulary. Genotype refers to the alleles that an organism has, and we use actual letters to represent this for the most part. And so here's an example. We have big B and little b. So usually dominant is big B, recessive is little b. Hetero means different, so 1 of each would be heterozygous. Homo means the same, so 2 of each would be homozygous. Phenotype is the actual appearance the trait an organism actually shows. So brown eyes versus blue eyes. In this case, brown eyes are dominant to blue eyes. And so you would have, as a reminder, brown eyes showing for big B, as well as for big B, little b, for the heterozygous trait. Okay? So an allele is a form of a trait. Genotype is the letter combination of alleles. Phenotype is the actual trait itself, what is displayed. In normal Mendelian inheritance, the dominant trait will be expressed if it is present. The only time a recessive trait is expressed is if there's 2 copies. Again, this is Mendelian inheritance. Now, the law of segregation says that during meiosis, each gamete only receives 1 allele for any trait. Now, there are errors in this. We talked about non-disjunction during the last screencast, and so this is assuming that there are no chromosomal abnormalities going on. Organisms produce an equal number of gametes with each allele. They have the same number of chromosomes, they have the same number of genes on each chromosome, and thus the same number of alleles. So this is what the law of segregation says, okay? Independent assortment says that the segregation of unlinked alleles have no influence on each other. And so since we have multiple alleles, as long as they're not on the same gene, they are just randomly assorted together when those gametes are formed. Now, there is a difference. If they are on the same gene, if the 2 different traits are on the same gene, then there is, they are considered linked, and we'll talk about that later on. But if we're talking about unlinked alleles, they don't influence one another. It's completely random how they're assorted out. And so either in regular biology or in genetics, you probably did what's called a dihybrid cross, where you had a couple of traits. The classic example is looking at peas, and we have green versus yellow, and we have wrinkled versus smooth. And they all independently assort out, and they give us this nice, beautiful box, 16 different boxes here. And then we can predict how many of each are going to be present. And we also find that we will have what's called a 9-3-3-1 pattern. Oops, I'm going to pause this here for a second and I will be coming right back. Okay, sorry about that. I had to take care of a kid that was getting ready for bed. Anyways, now we're looking here, there's going to be this proportionality. If we're looking at two dominant recessive traits, two different alleles, two different genes with different alleles, and we are looking at heterozygous crosses of them, we come out with this 9331 proportionality. So some of you may be familiar with doing a dihybrid cross and looking at the phenotypes and seeing that there will be, 9 showing the dominant of each form, 3 showing dominant of one, recessive of the other trait, another 3 showing recessive, the flip-flop, 3 showing the recessive of the first trait and the dominant of the second trait, and then 1 showing the recessive form of each. Knowing that is actually going to be a lifesaver because that shows that you have some kind of memory understanding of how to calculate probabilities in genetics. If you don't, that's okay if you don't know what I'm talking about, because we have some practice problems that will help us go through. And so let me go back to the present mode here. So here's the math skill. So I'm just going to go through. There's not practice problems on this particular slide. I've eliminated them, but we will be doing practice problems in class. And so just a little bit of a note here that chromosomal basis of inheritance provides an understanding of the pattern of passage or transmission of genes from parent to offspring. What that means is that there's a mathematical way of calculating the inheritance. Just as a review, sort of, that when these alleles independently assort, and thanks to the law of segregation, are in their own gametes, when they come together, chance has no memory. When these segregated alleles come and combine, it's all random. For each offspring, it's going to be a completely new combination. It doesn't repeat from the time before. It doesn't say, oh, we've already had big P, so we're not going to use it in this offspring. Each time it's just random chance. To calculate the probability of a phenotype occurring when you have multiple alleles, there's 2 different formulas that you'll want to know about. Okay, the first is the inheritance of either or, one trait or another trait. So to do that combination, what you do is you take the probability of trait A and you add it to the probability of trait B, and that will give you the overall probability of either A or B occurring. If you want to see the chance of them happening simultaneously within the same organism, you take the probability of A and you multiply it by the probability of B. So if you see the word and of smooth and green, you would multiply the 2 probabilities together. If you want to see one or the other, what are the chances of it being either smooth or green, you would add the probabilities together. The work done by Gregor Mendel investigated these simple dominant recessive relationships, and that's what a lot of genetics was focused on. When you start talking about many traits, you have a more complex inheritance. And in fact, even more recently, it's not just the number of traits that makes it complex, but we are finding out that it's not always a case of dominant recessive. It's not just a case of one allele having masking another allele. We have environmental effects. We have effects from different genes. We have effects from different proteins, and we'll get into all of that. In all cases, these complexities alter the ratios of offspring. The formula that was on the previous slide is just for simple Mendelian genetics. One common example of non-Mendelian genetics is incomplete dominance. And so these first couple of slides are probably ones that you covered in biology. An incomplete dominance, what it means is neither one of them are completely dominant, and your heterozygous will display a phenotype somewhere in the middle of the two. And so if we had white, WW, red, RR, and we have pink right here, it would be RW. And I think I actually have that. Yep, I on this slide right here. I forgot I had this one in here. And so here we have true breeding red flowers, so they're homozygous. True breeding white flowers, homozygous. The F1 generation will be heterozygous. They will display the pink. Now, F1 means the first filial generation. F2 is when you take and cross two of these first offspring. So in this case, for incomplete dominance, you will see that 100% will show the incomplete phenotype. If two of these F1 generations were to cross, though, you will see that 25% are red, 50% are pink, 25% are white. So 50% will show the incomplete heterozygous form, 25 homozygous red, 25 homozygous white. In codominance, there are two or more alleles that are dominant in a phenotype. So they don't show and display an intermediate, they both show. So in this case, instead of R and W making a pink, red and white, making a pink, you'll notice that red and white are both displayed here. This is kind of off a little bit, but here you have the red and the white both being displayed on this cow right here. Okay. You also have instances where you have multiple alleles that affect a phenotype equally and separately. It's not a blended phenotype. Instead, they just have different influences. Human blood typing is an example of this. In this case, we have three alleles, A, IB, and little I. AB are codominant, meaning that if you have blood type AB, it's going to show up as AB. If you have big A, it's going to show up type A blood. If it's big B, you have type B blood. However, if you have big B, little I, you have B, big A, little I, you have A, and I can't see, it's not showing down there. Darn it. Okay, and if you have little I right here, little I, you have O. Okay. And so you see you have your A antigen, B antigen, both antigens. Then we also have another factor going on here, and we have group O. We also have polygenic inheritance. And in this case, again, you have more than one allele working towards a phenotype, but they're not both going to be displayed. They add on to one another instead. They affect one another. They're not independently going to show. And so skin color, hair color, these are examples of different traits that are polygenic. Anything that shows a continuation and not discrete separations are probably good indications that they're polygenic traits. Skin color, height, weight, intelligence, and it's just additive. It's how many genes, how many alleles are responsible for that trait, and how many dominant recessive are available for each of those. So the previous examples were considered still Mendelian examples, polygenic codominance and incomplete dominance, although they were pushing the boundaries. Now we're going to get into even more complex patterns, and we're going to look at non-Mendelian inheritance. Many traits are not inherited according to Mendelian principles, so it's not inherited due to the law of segregation and independent assortment. Mendelian ratios only refer to situations where each gene is inherited independently of each other. So even though they're expressed and you have multiple alleles being expressed on the previous ones, like in codominance or incomplete dominance, each of those are inherited independently of one another. Now we're going to talk about when genes are linked and when there's a combination or an example where a phenotype probability will not be determined by necessarily doing Mendelian ratios, and instead we're going to have to look at the linkage to see if to see how common it's going to be. So before we get into that, we'll talk about a common linkage here, sex linked. Sex linkage refers to the inheritance of traits on sex chromosomes, and it was first investigated with the Drosophila fly, the fruit fly. So discovery of sex linkage occurred when we had a true breeding red-eyed female with a true breeding wide-eyed male. And when we say true breeding, that means it's purebred. It's pure. It's got a homozygous genotype for that. When these combined, the F1 generation, we had 100% of red-eye offspring. Okay, that would suggest that the red-eye offspring would be dominant. And that would suggest that when we had normal Mendelian ratios, the F2, if we were to cross two of these, would have a 1-to-1 ratio. However, we find that 100% of the females are red-eye, and 50% are red-eyed males of the males, and 50% of the males are white-eyed males. So instead of having a 1-to-1 ratio, or if you want to look at it 25%, 50%, 25%, regardless of the sex, we notice that there's two different probabilities depending on the sex. So sex does matter in this case. And the reason being that genes are on sex chromosomes. Now, the autosomal chromosomes, 1 through 22 for humans, and all other organisms that are also going to have autosomes, the number depending on what the organism is, have genes other than just the gender. The first discovery was by T.H. Morgan at Columbia University. It was studied with fruit flies, which are a common model organism because of how fast that they breed and how easy it is for them to be examined, and the short number of chromosomes, 4 chromosomes in total. In humans and other mammals, there are 2 sex chromosomes. There's X and Y. So if we have 2 X chromosomes, we have a female. And here's an example, the X chromosomes. If we have an X and a Y, we have a male. Now, in this case, what will happen is because we have 2 X chromosomes, we have gene redundancy. We might have 2 copies of an allele. In this case, though, because these are different different chromosomes, they're not the same chromosome, we're not going to have gene redundancy because the genes encoded for on the X are different than the genes encoded for on the Y. And you can tell that there are going to be a lot more genes encoded for on the X and very few genes encoded for on the Y based on the size. Okay. And so here you can see the actual cross from the one earlier with the red eye and white eye flies. And you can see how they came about here. We had on the X chromosome, we had our true breeding red-eyed flies with our white-eyed male. And since eye color is on the sex-linked trait on the X chromosome, it's going to be carried along as a recessive link to the X. If it's sex-linked, what we usually do is we write out the letter of the sex chromosome, either X or Y, and we write the allele itself as a superscript to it. Now, in the Y chromosome, there's few genes other than the SRY gene, and that's a sex-determining region. It's the master regulator for maleness, and it turns on genes for production of male hormones. That's essentially all that Y chromosome has with it. On the X chromosome, you have traits other than sex determination. You can't see because let me see. When there is a mutation on the X chromosome, it will often lead to a disorder that is displayed only when combined with a Y chromosome because there's no other X dominant gene to mask it. Hemophilia is one example. Here's hemophilia right here. And so if hemophilia, even if we have a carrier is someone that carries the disorder, in this case the lower H, but it's masked by the dominant form, the big H. So we have a female carrier and we cross it with a male that does not have hemophilia. That is normal for hemophilia. we find that we have the following results. We have a female that is normal, a female that is a carrier, a male that is normal, and a male that has just the one recessive form, which means that it's going to be displayed. The difference again between these two forms here is that the female has that additional allele on the X chromosome that's dominant that will mask this. Okay. So in this case, this is a carrier, this is disease or disorder, and the top two are normal. Here's another couple of examples. We have albinoism with a cat here. We have male pattern baldness. Both of those are sex-linked traits that are only expressed in one gender. The reason with a sex link that females don't display them is because they have it masked by the other X chromosome, the allele on the other X chromosome. It is important to note, though, that female mammals inherit two X chromosomes. We said this already. You get one X from mom, one X from dad. In order to prevent overexpression of genes, one X becomes inactivated during embryonic development. It condenses into a compact object or a bar body. And so what that means, and then this is random, whichever one becomes inactivated. And so depending on the one that is inactivated, you may notice differences in phenotype. A calico cat right here is an example. Calico cats can only be female. The reason is because in this black patch right here, what's happening is the brown fur, or I guess I should say orange fur, is inactivated. That's the X chromosome that's inactivated, the one with the orange allele. In the orange fur, it's the X chromosome that's carrying the black allele that is inactivated, and that becomes a bar body. And so depending on which one is inactivated, it's going to affect which fur color is actually displayed. Now, in order to track down the inheritance of these more complex types of inheritance patterns, we use pedigree analysis. A pedigree is just a family tree that describes the relationships of parents and children across generations. And so we can track down, it's like looking at a family tree to track down the inheritance of disorders. And so the keys that we usually use, squares are males, circles are females. If it's filled in, they're affected. And if it's halfway filled in, which doesn't have it on this one, if it's halfway filled in, they're a carrier. And so in this case, we are looking at widow's peak, and we have our first generation grandparents, and it's all based on what we can see, phenotype. These are the phenotype that's circled in. And sometimes we don't know what the genotype is, and we have to go through inheritance patterns and probability to predict and make decisions about what that genotype might be based on the number of offspring or the grandparents' phenotypes that were displayed. So in this case, is a widow's peak a dominant or recessive trait? Hopefully you see that it is a dominant trait, because right here, it's going to be expressed in either case. And you can see that it's expressed whenever there's a large one. Here we have attached earlobes. Again, we can follow the pedigree for it, and we see that it's recessive. We will look at inheritance patterns, and we will look at pedigrees. We're going to look at lactase prevalence, and we're also going to look at sickle cell anemia as two phenotypes, two disorders, to see how we can use a pedigree to look at probability and decide if it is a dominant or a recessive trait. So we'll get some practice with pedigrees, looking at that. Expect that sometime this week. There are a couple additional inheritance notes that I wanted to include, some terms that you'll probably come across, especially more recently in genetics discoveries. Again, this is just a another review of linked genes. We spent some time on the sex-linked genes earlier in this presentation, but it's not just sex-linked genes. Any kind of chromosome can have linked genes. It's any genes on the same chromosomes. Linked genes will usually be inherited together. However, it's not an always deal. Because of crossing over, there's chances that they will not be linked together. And so there's a way to mathematically determine like where the alleles are on the chromosome, how close or how far apart they are together, by looking at the gametes and the offspring that are formed and trying to calculate the frequency to see how close or how far apart the alleles are within the same chromosome. And so we'll look at map units, we'll look at some practice calculating frequency and how close in proximity on the chromosome a couple of different genes actually are. We also have something called non-nuclear inheritance. Now, nuclear DNA and nuclear genome, the DNA that we usually associate with human genomes, that's not the only DNA present that's going to be passed on. Chloroplasts and mitochondria also have their own set of DNA, and we talked a little bit about that with endosymbiosis as evidence for endosymbiosis actually occurring. And they are inherited and passed on as well. They are inherited entirely from the egg cell. And so when we're looking at mitochondrial DNA, we're looking at DNA in the chloroplast. We're looking at that egg transfer. It's all going to be from one parent and not the other. And so there are certain traits that are carried just on that mitochondrial DNA strand, and we'll look at that a little bit further on. It's going to be treated a little bit more like sex-linked for the purposes of determining probabilities. This is more of a recent development in looking at inheritance and tracking what traits and what genes are actually on the non-nuclear DNA. And so you may not see as much information in your reading as you do the other types of inheritance. Finally, we have environmental effects on genes that alter the phenotype. So phenotype is controlled. We've been talking about the alleles and the genes that are present, but also they can be controlled by the environment. And I know that this is kind of blurred together, but human skin is an example of a phenotype. The color of the skin is controlled by both genetics and environmental conditions. So if you're looking at getting a tan, for instance, you know that the color of the skin will change colors depending on the exposure to sun. And so you have one component that's genetically predisposed for your skin color, but it can also change within that. Hydrangeas are flowers that are influenced by the soil pH. And in fact, you used to throw out the tub water back in the old days, the wash water right out the window, and it would be more basic. And so you'd have these blue hydrangeas right there. And if you wanted them acidic, you'd have them in the front of the house. And so that's an example of a gene being altered by the environment. Sex determination in reptiles. The gender of an reptile, such as a alligator here, is controlled by temperature. The winter and summer coat of the Arctic hare, again, it's controlled by temperature. You have your brown fur color in the summer, and you have your white fur color in the winter. Again, here's a closer look at the tanning response in humans. And so these are all examples of the phenotype being altered by environmental effects. And finally, sometimes we'll talk about the plasticity of the genome, or you might encounter in your reading the plasticity of genes. And so different traits will interact with the environments in different ways. The ability of one genotype to produce more than one phenotype is what's referred to as plasticity. And so here we have 3 traits, and they do not change depending on the environment. There's no plasticity. In this case, we have red is definitely plastic. Purple is plastic. Blue is even a little bit plastic. It will change a little bit. In this case right here, we have highly variable plasticity, strong genotype by environmental interaction. And what that means is the slope is much greater. So we're going to have much more variation depending on the environment itself. And so that's just something to keep in mind. The effects of the environment on these genes is not consistent for all genes that we have on our chromosomes. Also, we have another term, and I didn't include it earlier. I could have put this further up, called pleiotropy. And so when we were looking at like human height and human skin color, and they were influenced by multiple genes that had an additive effect. Well, pleiotropy is affected by multiple genes, but they don't necessarily add on to one another. And so sickle cell, which we're going to explore a little bit further, is an example of pleiotropy. And then finally, the last term here, epistasis. It's when one masks the other. And so you will completely mask. So we have black fur color right here. It's going to completely mask the other fur colors when it's present. And so let me find a good example here. We have little b, big C. But notice that even though the big C is here to show brown, when the black is present and the black is dominant, it's going to completely mask the brown coloring. And so that's going to throw off the ratios a little bit. So I hope this kind of helps. It gives you some vocabulary. It gives you some examples. It doesn't go into very much detail. Use your textbook. Use your test prep book for that. Ask me questions in the discussion. I'll provide you with optional math practice and pedigree practice, in addition to the sickle cell and the lactase dominance that we have in populations activities that we're going to do. So use these resources, make flashcards if you need, but be cognizant that you will see terms and that you will expect it to give examples of these different terms. Okay. And I hope you guys email me with questions.